we have a very simple concept of infections in the lab. We work mostly with acute infections, and for us, in infections, first there is a microbe, then there is a barrier, and the barrier has to be broken by the microbe in order to start disease. Then it's very important that the microbe has to instruct the host cell what to do in order to produce pathogenesis. There is the initiation of inflammation, which is the most primordial innate immune response, and then in the vast majority of infections, there is resolution of the infection, meaning the microbe is destroyed. So this is our four easy steps to infection view of biology. And what I wanted to do today is to look at two aspects of these steps. The first one is the instruction of the microbe to the bocuse, and the second part, as Philippe already mentioned in his introduction, the resolution of infection. So, as many of you know, bacteria cause disease not because they grow in the body, but because they actually have molecules that specifically evolved to alter the behavior of host cells. And we work with Shigella, which we inherited from Philippe, but the truth is for several gram-negative bacteria like Shigella, Salmonella, Yersinia, and many others. And the way they instruct the host cell to do things is by injecting specific proteins. So this is a transmission electron microscope of Shigella flexneri. This is the cytoplasm, and you can see clearly the double membrane that covers the bacteria. You can also see these protrusions, which are the type 3 secretion apparatus. And the type 3 secretion is a machinery that delivers proteins from the cytoplasm of the bacteria into the cytoplasm of the host cell. And the scheme, so the dogmatic view of how type 3 secretion works is the following. There are effectors here in blue that have chaperones that deliver these proteins into the type 3 secretion. This type 3 secretion forms some kind of needle that traverses the two membranes of the microbe, and somehow they deliver proteins into the host cell. So this has been a very useful model to try to understand the system, but in fact there isn't that much direct proof that how this system actually works. So a group within my laboratory has taken a more biochemical view of how this works. So I took the liberty of taking imaging in a broader sense and to include the Armstrong resolution imaging. And what we were very interested is in the first step of this system, how are proteins delivered to the type 3 secretion and how they're inserted into the type 3 secretion so they can go through this very, very narrow needle. And the first approach we took is to do X-ray diffraction. So I'm sorry, I should go back one slide. There are two names you'll have to remember from the difficult bacteriology nomenclature. One is IPGC, which is the chaperone, and the second one is IPA-B, which is the effector protein. We purified IPGC to make crystals. This is a difficult and very medieval approach. It's always trial and error. It ended up that the wild-type IPGC cannot be purified. We design a mutant and loses its 4 last amino acids that can be purified and crystallized. This is a mutant that can complement the deletion of IPGC. So we believe that the crystals we get actually reflect the biologically relevant structure. So these crystals they are obtained in a kind of a random way. When they diffract, they are very useful. This is actually the first imaging we got from the crystal of IPGC. This is obviously not interpretable, so one has to create models to see how the protein actually looks. And this is how IPGC looks. And this is kind of an interesting protein. It is all alpha helixes, as you can see. And it is made of these things that are called TPR motifs. TPR motifs are very used in nature for many different effects. It's an effective construction model. You can also see that this is a dimer, which is very interesting, that I'll talk a little bit about later. One dimer is here in blue, and the other monomer of the dimer is here in brown. And what is interesting is that the dimer forms kind of like a hook. You can see the groove here. If it would be like a hand, like grabbing things, and the other monomer is actually hooked in the other way. So this alpha helix is inserted into the back of the second monomer in order to maintain the dimer in solution. We were able to show that this is actually a dimer by different spectroscopy methods and ultracentrifugation, and there are 3 specific links of this alpha helix into the other monomer that if we mutate, then we lose the dimer and we lose the activity of the protein. So the dimer form of the protein is absolutely essential for the function. This monomer, this dimer, as I said, this is the same image I showed you before. This is one groove that is formed by IPGC, and when we turn it 180 degrees, now we see the front of this groove here that makes the second part of the dimer. So we were able also to make the crystallized IPGC together with peptide from IPA-B. So actually the work of Claude Parseau defined genetically the region of IPA-B that is needed to bind to IPGC. And this is the amino terminus of this peptide. Actually, there is very few amino acids that hang out in the amino terminus of IPA-B, and this is a very large protein. IPA-B is around 60 kilodaltons. So there are several things that are interesting in this co-crystal. So first of all, let me orient you. This is the concave form of the groove of one of the monomers. This would be kind of like the form of my hand looking at you. And this is the IPA-B peptide that is hooked to IPGC. And what is interesting is that IPA-B is in the extended conformation. At least the section that binds to IPGC is in the extended conformation. There is a little area, I already mentioned, of IPA-B here that we suspect is the area of IPA-B that is required to engage into the actual type 3 secretion. And we think that IPGC is actually conserving the protein in an extended conformation to allow the insertion into the type 3 secretion. It is also interesting that this funny dimer that IPGC, this asymmetric dimer that IPGC forms, allows the binding of IPGC to IPA-B in two different directions. And actually, we don't understand at all if that has any functional meaning, if the two directions of IPA-B, of the two IPA-Bs that are linked each to one of the monomers of IPGC, has any functional significance. Another thing that we don't know about this protein is that actually work that Robert initiated when he was in Philippe's lab. IPGC is also the chaperone for IPA-C. And we don't know what is the binding site for IPA-C, and actually there is no link between the binding sites in IPA-B to IPGC and IPA-C to IPGC. So one of the interesting things about this structure too is that there are only 3 points where IPA-B is actually linked to IPGC. This we name P1, P2, and P3. And these are 3 amino acids that are in IPA-B that are absolutely conserved in all the IPA-B homologs in Salmonella, Yersinia, et cetera, et cetera. So evolution has absolutely conserved these binding parts. It is also these pockets that are formed in the chaperone in P1, P2, and P3 are conserved. And they are also very low interactions. They are in the micromolar KD range. And the reason we believe this is that as any chaperone, the protein has to bind, but also release the protein very easily, and we were able to measure the binding of IPGC to IPA-B by using calorimetry. So we believe now that this is the first step in trying to understand how the proteins are delivered to the type 3 secretion and how they can be hooked. So the next step is try to see how they are actually hooked into the machine to be transported to the other site. And another project that we think is interesting in this type 3 secretion research going on in the lab is PRGI. So now we're switching to Salmonella, but this is probably true for all the type 3 secretions, and they turn out to be homologs. Actually, the structural biologists don't think in terms of bacteriological species, but of protein species, and the proteins are very similar to Shigella, Salmonella, and Yersinia. MXIH would be the equivalent in Shigella. So we were able to crystallize PRGI by X-ray diffraction, and actually this was not great news because this was already reported for MXIH, and you can see it's also an alpha helical protein that it forms a hairpin that is actually fairly unspectacular. Our X-ray crystallography, like the X-ray crystallography of MXIH, lacked the amino terminus of this protein, so we were actually lucky to start a collaboration with the Max Planck for Biophysics in Göttingen, and using NMR technology, we are able to solve the complete structure of this protein. And it turns out that the rest of the protein was a continuation of an alpha helix with a small kink. The good thing about NMR technology is that you actually see the structure of the protein in solution, which is obviously more relevant than the X-ray crystallography. The problem of NMR technology is that you need enormous amounts of protein to solve any of these proteins. Also, the resolution of NMR microscopy spectroscopy is pretty low. You can only see proteins that are fairly small. Now, what I think is interesting about the mechanism of how actually type C secretions work is to understand how a monomer like this can actually build a needle. And for that, we are starting to use a very novel technology that is called solid-state NMR. And in solid-state NMR, one can actually see the protein when it's polymerized. So we were able to compare the NMR spectroscopy of PRGI inside needles and PRGI in solution. And this is a complex structure that I won't pretend I really understand. But what you see here is that there is a superimposition of the two spectra. And what is interesting is that there are isoleucines here in different places that have shifted from one of the structures to the next. And that means that that isoleucine has a different conformation in this insoluble form than in the solid state form. So we actually believe that what is happening is that there is a part in the amino terminus block that changes conformation in PRGI to allow the polymerization of the protein and the formation of the needle. So this is the examples I had at the Armstrong resolution of imaging of infections, and also the example I had for the instruction of the microbe into the host and how to behave. Now, if we go into the resolution of infection, we are moving in completely different scale. This is normal microscopy, optical microscopy, and also in terms of infection, we are at the end infection. So bacteria are very good at eliciting inflammation. The first cells of the host that arrive to the site of inflammation are the neutrophils, and neutrophils are very effective in killing microbes. Now, neutrophils are a little bit hard to work with because of several reasons. The first one is that there is no good animal model for human neutrophil biology. Neutrophils are slightly different than human neutrophils. Second, there are no cell lines to really try to understand neutrophil biology. So all the work that we have to do with neutrophils is with humans, with cells isolated from humans. So we bleed cells, we bleed people in the morning, and then we isolate neutrophils by density centrifugation. This is a very effective process. That means that when the purification is finished, we have cells that are around more than 98% pure. The other limitation of neutrophils is they are cells that are very short-lived. So they live around 6 hours in circulation, and if they are not called to a site of infection, they are disposed by the spleen. If they are called to a site of infection, they live a little bit longer, but not much longer. In vitro, we can make them live up to 12 hours, but not much more. And we're actually suspicious of experiments that are longer than 6 hours. So neutrophils have been well understood for many years. And this is a movie that we did with a special microscope that we have in the institute that is called a Richardson microscope. And they have a proprietary technique that actually is a diffraction mechanism where the cells are illuminated, and the diffraction consists that when they hit a lipid, they are diffracted. So that this is very useful for neutrophils because then we can see very clearly all the different granules that are present in the cell. So neutrophils have a nucleus that has a very unusual shape. They are called polymorphonuclear cells because of this funny nuclear shape, and they contain different granules. You can see that the neutrophil is very chemotactic towards bacteria. It is very effective about eating up the bacteria by phagocytosis. Then they engulf the bacteria in vacuoles, and as you can see now, this is engulfing this bacteria in a vacuole, and very shortly afterwards, the granules surrounded the vacuole, and then they fuse with the vacuolar membrane to create a very active phagolysosome. And neutrophils are very rich in different enzymes that have antimicrobial activity, like cathepsin G, neutrophil elastase, proteinase 3. They have many antimicrobial molecules like defensins and BPI and many others. And also inside this vacuole, they are very effective about assembling DNA-DPH oxidase, which is an enzyme that can convert oxygen to superoxide. And superoxide and the derivative of superoxide that we call radical oxygen species are supposed to contribute also to the antimicrobial effect of neutrophils. So this image is really not very different, although prettier than what Mechnikov observed more than 120 years ago, that is that phagocytes have the job of eating microbes and destroying them there. And what we observed is by activating neutrophils with microbes, we observe a new form or a new mechanism by which neutrophils can actually kill. And Philippe is right. This is all a story that is based in observation, in microscopical observation. So this is a scanning electron micrograph of the same type, of the same neutrophils that you saw in the movie. And you can see, for example, this neutrophil, or most of the neutrophils are very flat because they are activated and they are looking for microbes within the substrate. And they have many philopodia because they are always looking for things to eat. They are very phagocytic. And the observation was these extracellular structures that are outside the membrane that we call neutrophil extracellular traps, or nets for short. These nets are, as far as we know, only made by granulocytes, and that means neutrophils and other people have shown by mast cells, which are cells that are very closely related to the neutrophil in hematopoietic development. And these neutrophils have an unusual composition. So this is a classical immunomicroscopy, and this is a confocal image, and you see the projection, the Z-stack projection. And what we found is that we took elastase as a good marker for granular proteins. You can see in this neutrophil that is not activated that the granule localizes exclusively to the granules. I'm sorry, that the elastase localizes exclusively to the granules, but when the nets are formed, it also localizes to the nets. We also took antibodies like histone 2A and a marker for DNA. In this case, it's Hoechst. And you can see that histones and DNA are in the nucleus exclusively in a non-activated neutrophil, but they also localize to the nets. So these nets are made out of chromatin. I should say that we tried antibodies against different histones, against different granular markers. And now we know by a proteomic approach that actually there are other proteins that are cytoplasmic that are also present in the nets. And we actually don't understand how proteins are selected to be in the nets. And in vitro, it looks that the nets are hooked to the neutrophil that made them, and then they are very dense here close to the neutrophil that made them. And as we go far away, then the chromatin gets more ramified, and we can see the nets in more detail. So we took exactly an area similar to this, where the nets are very spread out, and did hand-resolution scanning electron microscopy to try to understand how they are formed, or how do they look. And what we observed is that there are these cables. Like you can see here, this is the most primordial image that we can see. These cables are around 50 nanometers in diameter, and we observe that it's rare to find these naked cables. Most of the time they are covered by these glomeruli or globuli that make around 50 nanometers in diameter. And some of the part is also actually covered by other structures that are more complex like that, and they usually stick to each other, like you can see here, and make very, very thick bundles. So in order to try and to start to understand how these proteins are distributed in the nets, what actually did, and all the work that I'm presenting in microscopy is really the work of Volker Brinkmann, who is the head of our microscopy core facility, is that he did a technology by transmission electron microscopy, he extended the nets, and then he did immunoelectron microscopy. And it turned out that for this detailed resolution, if we used gold particles to do immunoelectron microscopy, the gold particle distribution was not even. It probably is because of actually the weight of the gold that these things don't distribute. So this is not the classical gold immuno-EM that we are all used to, but actually the marker are quantum dots. The quantum dots are much lighter, so what we do is we hook the antibody, the specific antibody, to the quantum dot. These things are probably, because they are lighter, their distribution seems to be much better. So in the upper panel, we used an antibody, a monoclonal antibody against neutrophil elastase hooked to quantum dots, and you can see that the distribution of elastase is not equal throughout the net. There is more concentration here in what we call these globuli that are bigger. But when we took an antibody that actually we got from Mark Monestier at Temple University that recognizes an epitope that is formed by H2A, H2B, and DNA, then we can see that the histones are more equally distributed towards the net. So this has led us to the model that the chromatin somehow unfolds, probably because of the destruction of H1, which is the histone that links the different nucleosomes in chromatin, and then it's decorated by these extra proteins that make the nets. We confirmed that these granular proteins are present only in the bigger globuli by another kind of special technique, which is immunoscanning EM. And in this technology, we use gold markers, in this case, hooked to elastase antibodies. And then what Volker does is he illuminates with two different beams in order to get the gold particle and the structure of the nets, and then he pseudo-colors the images to be able to superimpose them. So what is in yellow here is the gold particle that marks the presence of a neutrophil elastase, and in the red background is the actual structure of the net. And again, you can see, for example, here, this primordial cable that is only around 50 nanometers in diameter, and in these more glomerular parts of the net, we see the marking for neutrophil elastase. So the image that is emerging from these nets is that they are, again, unfolded chromatin where different proteins somehow get attached to specific areas of the nets. We don't think there is periodicity to these glomeruli, but we'll have to look at more samples to do that. What there is periodicity for is for this distribution of nucleosome. So this is very indirect evidence. This is a net that we were lucky to get in a very straight form. And when we looked at, we measured these repetitive units in these very thin cables, we see that the repetitive unit is around 4.3 nanometers in diameter and around 10 nanometers in height. And this corresponds well to the published structure of the nucleosome, which is 5 nanometers in width and 10 nanometers in height. So this is another evidence to think that the chromatin really forms the core of the nets. Wanted to understand, or we are still very much interested in understanding that mechanism that allows net formation. And this is another question that where we decided to imaging microscopy to answer this question because of the limitations of the life of these neutrophils and the fact that they come as terminally differentiated cells into circulation. We cannot do any molecular biology. We cannot transfect them. We cannot transduce them. We cannot put siRNA, et cetera, et cetera. So in order to ask the question whether the neutrophil has to die to make nets, we use video microscopy. And the experiment was done in the following way. I'll show you 2 slides explaining the mechanism, and then I'll actually show you the movie. So we took a culture of neutrophils and activate them, but the neutrophils were stayed with calcein blue, which is a marker for living cells. So if in the presence of calcein blue, the cells turn blue. If they die, they lose the color. The second marker in this movie is annexin-5, and as Robert already explained, annexin-5 recognizes a specific lipid that is in living cells is exclusively the inside, so is inaccessible to annexin-5, and only when the cell dies it will be exposed to the surface. One of the tricks of the movie is that annexin-5 is in a very low concentration, so it's under the detection limit of the microscope, so you can actually not see green. And only when the cell dies, this lipid is exposed, and annexin-5 is concentrated on the cell, and the cell turns green. Now, in order to see the net, there is also a third and last marker, which is an FAB against, in this case, in the movie that I will show you, is against H2A, H2B, and DNA, but we've done similar films with anti-elastase antibodies and get very similar results. So again, this FAB is marked in red, and it's also at a very low concentration, so it is under the detection limit of the microscope. You cannot see it, and as soon as the cell makes nets, this chromatin is exposed, the antibody is concentrated, and we can see the nets. So this is actually the movie. And you can see the neutrophil here is blue, so it's alive. They go through this process where the nucleus first contracts and then expands. The still is still alive, and it's blue. And then after around 3 hours after activation, the cell undergoes a last contraction, and then at the same time, the cell loses the blue and becomes yellow and green. So this means that the neutrophil has to die in the moment of expulsion of the nets, and we think that the process that goes behind, where we see first the contraction and then the expansion of the chromatin, is when the nets are formed intracellularly. So the significance of the nets is a question that we are very interested in answering, and people have observed them in different animals and in different infections. And one of the questions we had is fungi. So neutrophils are extremely sensitive to activation by fungi, and fungi are one of the best inducer of net formation. And actually, fungi are not very susceptible to phagocytosis. So this is a scanning electron microscopy of a culture of neutrophils that was infected with candida, and the candida immediately made hyphae. This is one hyphae, and this is enormous, right? This is just a part of the hyphae. This would be much bigger than this room. And you can see the poor little neutrophil is too small to actually engulf the whole hyphae. So fungal infections in healthy individuals are actually quite rare, so it means that we have good defense against fungi, and we propose that maybe the most effective anti-fungi weapon of neutrophils would be the nets. And this kind of started making sense when we observed that one of the surprise molecules that is in the nets but is not in the granules but in the cytoplasm is calgranulin. Calgranulin is a zinc chelator that has a very strong anti-neutrophil activity, anti-candida activity, and actually it was not understood how calgranulin is actually delivered to the microbe because it's a cytoplasmic protein. So we are proposing that the net formation is the way to deliver calgranulin to the microbe. And this is a few last images on actually histology that I think is essential to understand any infection. And this is a pulmonary infection with candida. And you can see here the bronchus, and you can see this inflammatory site where you see in this case the section is stained with antihistamine antibodies, and you can see this extracellular material that is not in the nucleus, that is the size of nets. And if we stain with myeloperoxidase, which is a bona fide net marker, it's also present throughout the nets. And if we stain with calprotectin or calgranulin, this protein has many names, it's also present in the net. So when we superimpose all the structures, it looks like at inflammatory sites. You can see the nuclei of the cells. Many of these are neutrophils. You can tell by the shape of the nucleus. But also you see this extracellular fibrillar material that is reminiscent of what the nets should look like. And we can also take these paraffin sections and look at scanning electron microscopy to see the net. So this is not the same, but a similar view as the one I showed yesterday. Again, to orient yourself, this is a long infection with Candida albicans. This would be a bronchus, and here is a heavily a site that is heavily inflamed. And if we focus on this area, you can start seeing the structures of the candida. You can see here a yeast form that is starting to make a hyphae, and many other yeast forms that are present in the lung. If we look even in more detail, you can see the yeast form completely covered by these structures that have the right dimension and the right structure to be nets. And actually, when we take calgranulin knockout animals and infect them with albicans, they are extraordinarily susceptible to them. And we think it's because the nets of calgranulin knockout animals are not effective against net infection, net protection. So to summarize, in our 4 easy steps to infection, I showed you that microbes can instruct host cells to do whatever they want through the type execution apparatus. We believe that imaging will be essential. Imaging, again, in the sense of anything that allows us to see things, including X-ray crystallography, NMR spectroscopy, and solid-state NMR, to understand how this mechanism works and how type 3 secretion actually can deliver proteins from the cytoplasm of the bug into the cytoplasm of the host. And I also talk about a new mechanism on host resolution, which is the formation of net by a novel form of cell death that we believe is important in many different infections. So let me thank the people who actually did the work. So first of all the NMR was done in collaboration with the department of Christian Glisinger in the Max Planck Institute for Biophysical Chemistry in Göttingen. All the structural work is in the group of Michael Kolbe in my department, and Ravi and Michele were very important, where solved the IPGC structure both as a dimer and as a co-crystal with IPA-B. Omer solved the structure of PRGI. Konstantin was a postdoc in my lab who did all the work in Candida infections of mice, and is now in Umeå in Sweden. And finally, all the microscopy was done by Volker Brinkmann, who is an extraordinary microscopist and a very good scientist and an integral part to this whole net story. Thank you.
Thank you very much, Artur. Are there any questions?
Thank you. I was just wondering if other cell types might make nets upon dying, because it would seem once you have shown it, a sort of almost natural way of dying.
I certainly hope it's a natural way of dying. So if we take neutrophils, right, and we induce them to undergo apoptosis, for example, by activating the Fas ligand or just by letting them age, they don't make nets. They go through the bona fide apoptosis where the nucleus is condensed and there is DNA fragmentation. If we induce necrosis by incubating cells with pore-forming proteins, then the cells die, but they don't release the DNA, and there is not this mechanism where the DNA is combined with specific proteins from the granules into the cytoplasm to make nets. It's true. If you take a HeLa cell and you put it in water, it will release its DNA. Right? But it won't be in combination with specific proteins of the cytoplasm. Right? Now, we've tried very hard to look for other cells that make nets. Well, not very hard. We've tried a few experiments. Macrophages would be an interesting cell. Macrophages, as far as we know, cannot make nets, but this is a negative result, and until maybe we didn't find the right stimulus. Mast cells and eosinophils do make nets. Other people have shown that.
Arturo, I was really fascinated by the PRGI stuff you have been doing. The question is, are you actually able with that to also follow if there is an impact of the tip complex on the needle, or can you also use that to look at the length of the needle and such sort of things?
Yeah, there is you mean the tip of the complex is IPA-D in Shigella?
I mean, in Shigella, it's IPA.
Yeah, and the IPA-D, we are in the process of solving too. And we don't understand, that would be one of the questions that we would like to answer. If we could make a co-crystal between PRGI and IPA-D, or the equivalent in Salmonella, Shigella, I always get them mixed up, that would be an interesting way to see how is the length regulated, but we don't have those crystals yet.
Arturo, are these nets induced by the bacteria? I mean, if you take a normal commensal bacteria, will it induce it, or is it something that is related to Salmonella?
No, we think it's not pathogen specific, it's bacterial specific, and we can do things, for example, if we take very pure LPS, the production of nets is very poor. If we take dirty APS or real endotoxin, we get better nets, but the best way to do nets is by incubating with microbes. And it doesn't, we've tried with Klebsiella, Shigella, Staph aureus, Candida, Aspergillus, they all seem to work.
So do you think
antibodies are very good at inducing that. Sorry?
Is it TLR mediated, do you think?
It's TLR mediated, but not required, because we can do, if we can do it through antibodies, right? Neutrophils are very rich in Fc receptors, so antibodies also induce nets. So we think there is several images, and we're actually not working on that, unfortunately, that come from the surface through receptors linked to PI3 kinase and protein kinase C that then activate the NADPH oxidase, and that is the requirement. But what happens upstream of NADPH oxidase, we don't think is anything special. It's like any other receptor.